One of the most important functions which are involved in the formation of urine is tubular reabsorption. And in this video, we'll be covering the three important compounds which are reabsorbed by our kidneys. That is, firstly the glucose. Then we'll learn about the reabsorption of sodium, and finally about the reabsorption of water. So to begin with, let's start with the parts of a neuron. So we know that a neuron has a glomerulus, which is a tuft of capillaries with an afferent and efferent arteriole, and then it has the Bowman's capsule, and these two segments are involved in the filtration. That is the filtration of plasma to produce urine. And then we have our proximal convoluted tubule, that is the PCT. The urine then passes through the loop of Henle, and this loop has a descending part, that is the descending loop of Henle, and it has an ascending part, that is the ascending loop of Henle. Afterwards, it moves into another convoluted curved tubule, that is the distal convoluted tubule, which is distal to the loop of Henle. And finally, they all join at the collecting duct, where the concentration of urine is completed, and finally the urine passes out of the kidney into the ureters, into the calyxes, and finally into the ureters and outside through the bladder and urethra. So let's now move on to the actual topic. That is, we'll discuss firstly about the reabsorption of glucose. So there are four parts, right? So our glucose can be reabsorbed at, the first part is the PCT, and then we know there is a loop of Henle, then there is the DCT, and finally we have the collecting duct. Now, how much of glucose is actually reabsorbed? 100% of the glucose that is filtered out by the kidneys are reabsorbed by the PCT itself. That is, they are reabsorbed immediately after they are getting filtered, and they do not actually reach the other parts, that is the loop of Henle, DCT, or the collecting duct. Now, what is the mechanism through which the glucose is reabsorbed? So for that, here we have our blood vessel, and this is our cells of PCT, and this is our tubular lumen, or the space of tubules. Now, how is glucose absorbed? Glucose is absorbed by an active mechanism. So we know that absorption could be via two mechanisms. That is, either there could be an active transport mechanism, or there could be passive transport mechanism, right? Now we know some examples. For passive transport, there is diffusion and osmosis. Whereas active transport, they involve the energy. So ATP involvement is there, involving the sodium, potassium, ATPase usually. And glucose is actually absorbed by the secondary active mechanism. So this is our glucose molecule, and they are absorbed by secondary active transport mechanism. Now, what is a secondary active transport mechanism? Here we have the sodium, potassium, ATPase, which will transfer sodium molecules from the PCT into the blood vessel. And now this actually creates a concentration gradient, right? Like inside the PCT, the sodium concentration has decreased. So as a result, what happens? What happens is that we have our sodium glucose co-transporter, and this sodium glucose co-transporter will actually transfer our glucose along with a sodium molecule. So these are our sodium molecule, and this is our glucose molecule. So the sodium glucose co-transporter transfers the glucose into the PCT, and this glucose is then carried out into blood by another transporter. Now, what are the names of these transporters? The name of the sodium glucose co-transporter is SGLT2. So there is the SGLT2, and the name of this transporter at the basolateral, this is the basolateral end of the PCT, and this one is the apical end. Now, the transporter at the basolateral end is the GLUT2 transporter, whereas those at the apical end is the SGL2 transporter. So to summarize, 100% of glucose is absorbed by secondary active transport, that is the sodium glucose co-transporter, or the SGLT2 at the apical end, and at the basolateral end we have the GLUT2 transporter. Now, we said that 100% of glucose is absorbed, right? What that also means is that there should not be any glucose in the urine, because it's completely reabsorbed. So if glucose is found in the urine, it indicates that there is some pathology in our body. Now, what could it be due to? It could be due to some defect in the reabsorption, because 100% should have actually been reabsorbed, but in some pathology or in some pathogenic condition, we are not able to reabsorb 100% of glucose. How or what actually happens in such a scenario? To understand that, we need to know the concept of transport maximum. In a normal individual, let's say that 5 molecules of glucose enters the tubules after filtration, And let's say that we have 10 transporters. So we have 10 transporters in the basolateral membrane. So what will happen? Very easily the 5 molecules will be transferred by the SGL2 because there are 10 of them, right? Now let's say that instead of 5 we have 12 molecules of glucose. What will happen now? In this scenario we only have 10 molecules of co-transporters. So what will happen? 10 of them will enter or will get reabsorbed, whereas 2 of them will pass through the urine. So now we have glucose in the urine. Now what condition causes excess of glucose in the tubular lumen? Excess of glucose in the tubular lumen means that excess of glucose is filtered by the glomerulus, which means that excess of glucose is present in the renal arterioles or in our blood vessels. So what is that condition where there is excess of glucose in our plasma? That is diabetes mellitus. That is diabetes mellitus. That is, in diabetes mellitus, what happens is that the plasma glucose level is higher, and as a result what happens is that the filtered glucose is actually higher than the capacity of our tubules to absorb the glucose. Now this ability, what do you call this capacity or this maximum ability? That is what we call as tubular maximum. So tubular maximum is defined as the maximum rate at which active transport can transport or reabsorb a substance. Or in other words, we could say that when TM is attained, what happens? When transport maximus is attained, we enter into a state of saturation. That is, all the transporters are used for reabsorption. So in diabetes mellitus, the glucose that is filtered is greater than the transport maximum. Now what is the normal value of transport maximum? The normal value is 375 milligram per minute in males, and in female it's 300 milligram per minute. So what does this unit mean? This unit means that every minute 300 milligram of glucose can be reabsorbed by the PCT when it works in a saturated, in the maximal state. Now then there is the other concept, that is renal threshold. To understand the concept of renal threshold, let's say that instead of having 10 transporters, let's say that I have here only 3 transporters. Okay, now in a normal state I said that 5 molecules of glucose are filtered through the tubule, right? As an example, what will happen here? Now since we only have here 3 molecules of SGL2, 3 of them will get reabsorbed, whereas again 2 of them will pass through the urine, right? What does this mean? Even though the glucose in our blood level is normal, we still find glucose in urine. Why does this happen? Here we will find that the defect here is not diabetes, where there is increased plasma glucose level. The glucose level in our body is actually normal, but here the defect is in with the transporter, that is SGL2, which means that this condition, it leads to a condition known as renal glycosuria. So the condition is termed as renal glycosuria. What it means is that glucose is found in the urine even though the plasma levels are normal. Now we need to know what is the value at which the glucose is found in an individual with normal blood glucose, and that value is what we call as renal threshold. Renal threshold is defined as the plasma level at which glucose first appears in urine. Theoretically, the value of renal threshold is said to be 300 milligram per deciliter. What it means is that when the plasma blood glucose level is 300 milligram per deciliter, only once it crosses that limit we'll find glucose in the urine. Otherwise glucose is absent in urine up to 300 milligram. However, in reality what it is found is that glucose appears in urine once the blood plasma levels reach 180 milligram per deciliter. Now why is this disparity? We have a disparity of 120 milligram, right? This is due to the concept of splay. Splay basically means is that our nephrons do not function equally and maximally, and as a result what happens is that there will be a difference between the theoretical and actual value. So the reason for this splaying is that our nephrons do not function equally and maximally, or in other words we can say that our nephrons are heterogenic in nature. So that's about glucose. Now let's move into sodium reabsorption. So when we speak about the sodium reabsorption, we need to know how much percentage of sodium is actually reabsorbed. Not 100%, unlike glucose, 99.4% is reabsorbed, which means that 0.6% is excreted in urine. Now once again, we have our PCT, then we have our loop of Henle, then we have the DCT, and distal to that we have the collecting duct. Let's begin with the PCT. In the PCT, how much percentage is reabsorbed? About 60% amongst the 99.4%, 60% is reabsorbed from the PCT itself. So what are the mechanisms? There are several mechanisms. At the early part, this is the early part of PCT. Now there are two mechanisms. The first one we had actually already discussed. That is when we discussed about the co-transporters. That is, we have, this is our sodium molecule, this is our glucose molecule, and we have the sodium glucose co-transporters which worked with the sodium potassium ATPase via the secondary active transport mechanism. So that is our first mechanisms which we had already discussed. Instead of glucose, it could also be amino acids or phosphates. So they have the secondary active co-transport mechanism. And then we have the bicarbonate secretion. So this is the first mechanism. And our second mechanism is actually with bicarbonate reabsorption. So sodium is reabsorbed along with bicarbonate. Now what happens here is that bicarbonate in the ionized form, it cannot enter into the tubular cell. So what happens is that it will combine with H plus and that will form H2CO3, which we know that by the action of carbonic anhydrase it gets split into H2O plus CO2. Now this molecule can easily diffuse into the tubular cell. So and then what happens is that this will again regenerate, or it will turn back into, it's a reversible reaction, again catalyzed by carbonic anhydrase. It will revert back into the bicarbonate form plus H plus. Okay, now what happens is that this H plus that we have here, this H plus must go back into the lumen, right? For that, what happens is that this H plus is exchanged with sodium. So we have sodium which is exchanged with the H plus. H plus ions move outside whereas sodium moves inside. That is in opposite direction. So this is an antiport. So the second mechanism, or the HCO3 absorption, is via the sodium hydrogen antiport. And this sodium, this is also again a transport mechanism involving the ATPase. So here what happens is that again we use the help of sodium potassium ATPase to enter into the blood vessel. Now this sodium molecule and the bicarbonate molecule, right? The bicarbonate molecule will enter into the blood with the sodium, along with the sodium. That is the sodium bicarbonate symport. So that's how sodium gets absorbed. These are the two mechanisms of sodium absorption in the early part. Now moving on to the late part of PCT. In the late part, there are two other mechanisms. One is with the chloride absorption. So there we said the bicarbonate absorption. Here there is the chloride absorption. The mechanism is actually somewhat similar. Here again we take the help of sodium H plus antiport. So sodium moves into the cell and H plus ion moves out of the cell. Now for chloride absorption, unlike that of bicarbonate, the chloride is reabsorbed from the lumen into the cell with the help of an antiport. And that is the chloride anion antiport. So one anion is exchanged with the chloride ion and chloride enters, and this chloride will enter into the bloodstream along with the potassium. So there is a chloride potassium symport or a co-transport mechanism. Now what happens to this anion? This anion must be recycled back, right? The first question is, where do we get this anion from? This anion will combine with our H plus. So our H plus and anion will combine and they will form a compound. Let's call it H anion. H plus, we know that neither H plus nor anion itself cannot enter into the PCT because they are in the ionized form, whereas H anion is in the non-ionized form and that will diffuse into the PCT. And then again they will dissociate into H plus and into the anion, and this anion goes with the goes into the reabsorption of chloride, whereas our H plus is excreted out into the lumen. The main mechanism here again, as I have said, for chloride absorption we also reabsorb sodium along with it, and the sodium, this antiport is a secondary active transport mechanism. Therefore we make use of sodium potassium ATPase. So let's write down the mechanisms. There are four mechanisms, and the first one is the co-transport mechanism with glucose, amino acids, phosphates, etc. The second mechanism is the sodium H plus antiport associated with bicarbonate absorption. The third mechanism is again the sodium potassium, hydrogen or H plus antiport with chloride absorption. Finally, there is one more mechanism which I forgot to mention. All of the sodium, so we have reached the late part of PCT, right? Around 67% of sodium is reabsorbed. As a result, what happens is that positive charge is lost from the lumen. So naturally what happens is that the proximal convoluted tube is going to be more positively charged compared to the lumen, which will have a net negative charge, right, because of loss of positive sodium. So here what happens is that there is going to be chloride passive movement. So there is going to be a paracellular chloride movement from the lumen into the blood vessel, and this movement of chloride is actually not through the channel, but it is paracellularly. Paracellularly means that between the two cells. So here we will have our second PCT cell. Now between these two cells, between the gap between those cells, the chloride ion will diffuse into the blood, and as a result what happens when the chloride ion goes when excess of chloride ions diffuse, we will again have a reversal. We'll again have positive charge on the lumen and negative charge into the side of PCT, and then what happens is that instead of the chloride, the cations will move, that is sodium, potassium, magnesium, and these cations will paracellularly passively get absorbed into the blood vessels. So that's actually a fourth mechanism. There is paracellular chloride driven sodium transport. So these are the four mechanisms of sodium reabsorption in the proximal convoluted tubule. Let's now move into the next segment, that is the distal convoluted tubule. So we know that the loop of that's the loop of Henle. In the loop of Henle, we know that there is a descending loop of Henle, and then there is the ascending loop of Henle. The ascending loop of Henle again is divided into two parts. There is a thin part, and then there is the thick part of ascending loop of Henle. Now in the descending loop of Henle, what we need to know is there is no absorption. It's an important point. There is only absorption of water. There is no absorption of sodium. It is impermeable to solutes, so nothing much to look over there. When it comes to the thin part, what happens is that here there is going to be a passive sodium absorption, which again is minute. Then we have the thick part. Here it is the thick part. Here we again have an active sodium absorption. And how much percentage? We said that in PCT 60% is absorbed, whereas here 25% is absorbed. So 25% of sodium absorption occurs in the thick part of ascending loop of Henle. Now what are the mechanisms? So we have a channel present on the apical side. This is the apical side, and this is the basolateral side of our thick ascending loop of Henle. And this channel is called as the sodium potassium 2 chloride. So it transfers sodium, potassium and 2 molecules of chloride into the cell from the apical side. This is again an active transport mechanism, and it uses the sodium potassium ATPase. And in addition to that, there are some other transporters as well in this region. On the basolateral membrane, we have a chloride CKB channel. Now moving on to the distal convoluted tubule. Here 5 to 7% of sodium is absorbed, and the mechanism here is really simple. Here we have another transporter, and this transporter is the sodium chloride co-transporter. This is again an active transport mechanism that makes use of the sodium potassium ATPase, and this helps in the reabsorption of sodium and chloride, and by this 5 to 7% of sodium absorption takes place. Now what if this transporter is blocked? This transporter is blocked by the action of a diuretic known as thiazides. So thiazides are diuretics which act on the sodium chloride co-transporter at the DCT, and as a result sodium reabsorption is decreased, and along with sodium water is not reabsorbed. It leads to diuresis, that is increased urine excretion. Now finally what we have here is our collecting duct. In all the remaining three parts, I have not seen any hormone which is getting involved. Whereas when it comes to the collecting duct, here hormonal absorption takes place. First point is that 3 to 4% of sodium is absorbed. That's our first point. Now coming to the collecting duct. Collecting duct actually has two types of cells. There is a P cell and then there is an I cell. Now it is the P cell which is associated with sodium reabsorption. And here the absorption, the important point is that it is associated, or it is due to the hormone which is known as aldosterone. So there is an aldosterone-dependent absorption of sodium. If the aldosterone is not secreted, there is not going to be any reabsorption of sodium from the collecting duct. So what happens? So what is the mechanism? Now aldosterone is a steroid hormone. The aldosterone that is produced from the adrenal cortex. From the adrenal cortex, the aldosterone hormone reaches into our collecting duct, that is the P cell of the collecting duct. And since being a steroid hormone, it can easily enter into the cell, and inside the cell it acts on the nucleus, and it causes two functions. Firstly, what happens is that it will increase the insertion of a special type of channels which are known as the epithelial sodium channels, ENAC, epithelial sodium channels. And secondly, what happens is that it will increase the synthesis of new epithelial sodium channels. So firstly, it will activate the existing ENAC, and in addition to that, it will also increase the synthesis of new epithelial sodium channels. So as a result, what happens is that there is going to be absorption of sodium into the collecting duct, and this sodium again it enters into the blood via the sodium potassium ATPase. Now what happens if we block the epithelial sodium chloride channel, the epithelial sodium channel? What happens is that if it is blocked, again sodium absorption is somewhat decreased, so there is going to be a diuresis again, and this diuresis is brought together by potassium sparing diuretics such as amiloride. We'll learn more about them in the pharmacology session. Diuretic, that is spironolactone. Spironolactone is more concerned with the action of aldosterone than the action on the epithelial sodium channels. So that's about sodium reabsorption. Having said that, now we move on to the water reabsorption. Again, we have PCT, loop of Henle, we have our distal convoluted tubule and the collecting duct. How much of water is reabsorbed? Like sodium, 99%, not 99.4, 99% is reabsorbed and 1% is excreted in urine. Now the reabsorption of water is actually really simple. In the PCT, it follows sodium. So whenever sodium is absorbed, water follows it. So the similar amount of sodium and potassium is reabsorbed. How much percentage of sodium was absorbed? 67, right? So similarly, 67% of water will follow the sodium. It's a solute solvent, by bond, you could say. That is, whenever sodium is reabsorbed, water will passively follow it. When it comes to the loop of Henle, however, there is a difference. We know that the descending loop of Henle and ascending loop of Henle has different permeability when it comes to sodium and water. In the descending loop of Henle, H2O is reabsorbed. It is permeable to H2O, whereas it is impermeable to sodium. Whereas in the ascending loop of Henle, it is the opposite. That is, there is no water reabsorption, whereas there is sodium reabsorption. Here 15% of water is reabsorbed. In the distal convoluted tubule and the collecting duct, however, water does not follow the sodium. Instead, it is dependent on a hormone, and that is the ADH, that is anti-diuretic hormone. Just like aldosterone regulating sodium reabsorption, the anti-diuretic hormone will regulate the water reabsorption in the late part of DCT and in the collecting duct. So what happens? Here we have our hypothalamus, and the hypothalamus, the posterior part of hypothalamus produces the ADH hormone, that is anti-diuretic hormone, and this anti-diuretic hormone, it acts on the V2 receptors that is present on the P cells. Again, the P cells of collecting duct. So in collecting ducts, we had said that P and I cells are present. The P cells are responsible for the sodium reabsorption and the reabsorption of water as well. And then what happens is that it's a G-coupled protein, G-protein coupled receptor sending secondary messengers which increases the CAMP. This is actually explained in endocrinology. And ultimately what happens is that there is going to be insertion of water channels, that is aquaporins 2. And these water channels are known as aquaporins 2. And with the help of aquaporins 2, what happens is that water alone without sodium is absorbed into the blood from the tubular lumen. So ADH dependent, aquaporin 2 dependent water, selective water reabsorption occurs in the collecting duct and in the late part of DCT. This is a really important mechanism. Why? What happens when ADH increases? When ADH increases, what will happen is that there is going to be an increased absorption of water from the collecting duct and from the late part of DCT. As a result, what happens? The final urine output is going to be concentrated, right? This condition is called as SIADH, that is syndrome of inappropriate ADH secretion. What happens if the ADH levels are decreased? In the absence of ADH, there is not going to be any absorption of water from the renal tubules. As a result, what happens is that there is going to be increased H2O excretion, and this will lead to diluted urine. This actually occurs in a condition, excess of urine is passed out as well, and this occurs in the condition known as diabetes insipidus. Why is ADH decreased? ADH could be decreased either due to the defect on the hypothalamus, because there is some defect in the posterior hypothalamus because of which ADH is not secreted, that is known as the central diabetes insipidus, or the defect could be in the kidneys, whereas there is some defect in the receptor, or ADH is not able to respond to the receptor, that is called as nephrogenic diabetes insipidus. So I guess that's about the absorption of 3 most important compounds, and that's it for this video.